All right, welcome everybody. These set of notes are on enzymes, and there's a little blurb in your book on page 57, but most of this is not in your book. The standard that we're covering here is to identify how enzymes control chemical reactions in the body. So a quick review of enzymes is that the definition is that they're proteins that act as biological catalysts. What this means is that they lower the activation energy and increase the rate of chemical reactions. There's a couple of facts that are true for all enzymes, and one is that they're specific to only one chemical reaction. And there's a very good reason for this. In class, we've talked a little bit about the chemical reaction that happens in your blood to convert carbon dioxide into carbolic acid. Now, the reason why that's important is because CO2 dissolved in a liquid like your blood makes it very acidic, and this is not so good for our circulatory system. So there's an enzyme needed to change that carbon dioxide over into something else that is not going to harm our circulatory system. And we want those enzymes to be functioning all the time. Yes? There are other chemical reactions that are controlled by enzymes, such as the replication of DNA, which is part of the cell cycle and the cell getting ready to split and make a new copy of itself. And the enzymes that are involved in replicating the DNA and helping the cell split, we don't want those on and running all the time. Because if our cells were replicating all the time, just it replicates and then again and again and again and again, this is actually what creates tumors on our skin or on one of our organs, and sometimes these tumors can be cancerous. So we don't want all chemical reactions running all the time. So to solve that problem, each enzyme is specific to only one chemical reaction. And we can turn these enzymes off if we need to, and we'll talk about that later in the notes. Here's another fact about enzymes that's not in your book. The enzyme's name is usually from the chemicals in the reaction that it catalyzes. So we'll take a look at that. So here are two examples of enzymes, and if you'll notice, both of these enzymes end in ASE. So ASE, whenever you see this ending, and the R may be part of it too now that I think about it. So you see this ACE, so carbonic anhydrase or DNA polymerase. This means that it's an enzyme. And what would you guess the DNA polymerase does? Well, it's involved with DNA somehow, right? Poly means what? More than one, right? So the DNA polymerase is involved in making more than one strand of DNA. So DNA polymerase is involved in copying DNA. Carbonic anhydrase. Okay, this one's a little bit tougher, but we know that it's going to involve what element? Carbon. So this is the one that helps turn carbon dioxide into carbonic acid in our blood so that we can survive. So whenever you see examples of chemicals or proteins, and they have this ACE at the end, the polymerase, anhydrase, they are enzymes. So let's take a look at a very simple drawing of an enzyme and talk a little bit about how it works. The enzyme in this picture is represented by the greenish-blue circle here, and the substrate is going to be this pink thing here. So what is a substrate? What is this pink thing? The substrate are simply the reactants of a chemical reaction. Now there can be one substrate, or there can be more than one. There can be 2 or 3 substrates involved in the chemical reaction. For our purposes, substrate and reactants, as far as the chemical reaction goes, you can use them interchangeably. They are a little bit different when you get into chemistry, but for our purposes, as long as those two mean sort of the same thing for you, that's okay. I would prefer you use the term substrate. When you're talking about the enzymes, this is the initial or the reactant that you have, and it's going to bind to the enzyme at the active site. And by bind, we mean it's actually going to attach with some chemical bonds that line up just right here at this active site on the enzyme. So the active site is the part of the enzyme where the substrate will fit into it. There are two types of chemical reactions, or there are two types that enzymes participate in. They can be synthesis reactions or decomposition reactions. The synthesis reactions, the word synthesis means to put together. So a synthesis reaction is when you have two or more substrates and they're combined. So think about that for a minute. A synthesis, you're putting something together. So the enzyme is going to be putting together two or more substrates to form one product. You can also have decomposition. You guys know what decomposition means? It means to break things down. So if the enzyme is a decomposition enzyme, it's going to take one product and create one or two or more products. So you start with one substrate and you break it down or decompose it into two or more products. Now, there are some things that can regulate enzyme activity. So how quickly enzymes perform their functions. Temperature and pH are the biggest factors, although there are a few others. These are the two that we're going to talk about. So temperature. Each enzyme has an optimum temperature. What this means is that it has the best temperature. So there is a temperature at which it will perform the most chemical reactions in a given time. If the temperature is too high or too low, it can denature the proteins in the enzyme. And we'll talk about exactly what denature means in a few minutes, but on the short, it means that it doesn't work anymore. So you can actually destroy an enzyme by having the temperature too high or too low. The same thing is true of pH. Each enzyme has its own optimum pH. So it likes either acidic conditions or basic conditions, or perhaps it likes neutral conditions. And again, just like with temperature, if you get too acidic or too basic, you can destroy the enzyme. Let's look at some graphs of this. Here's a nice graph that's showing temperature. So we have 0 degrees Celsius here on up to 60 degrees Celsius here, which is quite hot. On the left-hand side, we have the rate of reaction. So this is the amount of starches that are digested per minute. Right? It doesn't say whether these are grams but it's the amount of starch that's digested. So at 10 degrees Celsius, you have about 8, we'll call them grams, I guess, 8 grams of starches that can be digested in 1 minute. As the temperature goes up, you can get to a rate of about, it looks like 34 grams of starch per minute, and that temperature is about 40 degrees. So this right here would be the optimum temperature for this enzyme. It can function when it's colder and it can function when it's warmer, but it's just not going to be as efficient. If you'll notice on the right hand side, after 40 degrees, the efficiency drops off very steadily, and this is because above, it looks like at above about 50 degrees, this enzyme will actually become denatured, which means it's destroyed, and it can't function anymore, and those reactions will slow down if not stop. So let's look at these two graphs. The top graph is showing temperature of two different enzymes, and I'm showing you this because each enzyme is unique, and they all have different optimum temperatures, and they all have different optimum pHs. If you look at the optimal temperature for most human enzymes, that's this black line, they work best at, it looks like about 37 degrees Celsius, which is close to our body temperature. So one of the reasons that our body may work so hard to maintain our body temperature is that this is the optimum temperature for our enzymes to function, which is a very good thing. The green line is the optimum temperature for some of the enzymes found in those extremophile bacteria, those archaea bacteria that may live in very hot, those thermophilic, meaning they love the heat. So these bacteria can live at much higher temperatures. And to give you an idea, remember that this is about human body temperature, so 98.6 degrees. We're making a huge jump, so hundreds of degrees Fahrenheit here. And that's the optimum temperature for the enzymes in that bacteria. So each enzyme has its own optimum temperature at which it will function. The same is true for pH. So if you look at these are both human enzymes. The black line is representing pepsin, which is an enzyme that's found in the stomach, and its optimum pH is 2, which is a medium acidic. This is about what the hydrochloric acid, the environment in our stomach. The optimal pH for trypsin, which is an intestinal enzyme, is very basic. And you may not realize this, but our intestines have a very basic environment. So these two enzymes have different optimal pHs. So what that means is as your stomach is doing its thing, grinding up your food, digesting your food, as that food moves into your intestines, any of the enzymes that go with it, because it just sort of all moves together, any of the stomach enzymes that move into your intestines actually stop functioning and they become denatured because the pH is way too high for them. It becomes too basic of an environment. So pepsin, this enzyme in your stomach, will actually stop functioning once it moves into the intestines. And that means that the cells in and around our stomach are constantly making this enzyme to help us digest our food. So what can promote enzyme activity? How can you get an enzyme to work at its peak performance? The two we just talked about are to have the optimum conditions, like optimum pH and temperature, which is what goes on in our stomach and our intestines and things like that. The other one is to increase the concentration of the reactants or the substrates. So think about your stomach. You can get these enzymes to catalyze more chemical reactions if you put food in there, because suddenly you have food for them to break down. So more reactions are going on. Less reactions are going on when you're hungry and there's not very much food in your stomach. So if you increase the concentration of the substrates or the reactants, the things that the enzymes need, then you actually increase the performance of the enzyme because there's more stuff for them to work on. And that's what I mean by concentration. Enzymes can also be inhibited. Remember at the beginning of the lesson I told you that you didn't necessarily want some enzymes to function all the time. So there are actually chemicals called inhibitors. And these chemicals will move into the active site and kind of plug it up. Nothing happens. So in this case here, the green part, the green bubble is our enzyme. Our substrate is this pink and purple molecule, and when the pink and purple molecule attaches to the active site of the enzyme, which would be this right here, the enzyme will break them apart. So this would be a decomposition reaction because you have one substrate and it's being broken into two products. So this decomposition reaction happens when the substrate attaches to the active site here. To inhibit the enzyme, to prevent it from creating these products. Perhaps this product is something that we don't need in our bloodstream at the moment. So we don't want this enzyme to continue to break apart the substrate because we don't need these right now. Our cells will produce more inhibitors, and so the more inhibitors there are, the more likely that they're going to sit in the active site. But because they only sort of fit partially, you see this isn't this part of the active site isn't filled, then the enzyme just doesn't work, and the inhibitor kind of sits there, and then it bounces out, and then another inhibitor may sit in there, or perhaps a substrate may find its way there. Either way, it depends on the concentration. If there's a high concentration of inhibitors, then you're not going to get many of these products because the inhibitors are going to take over the sites. If you have a high concentration of substrate and a very low concentration of inhibitors, then the reaction will continue to run and you'll get the products that are needed. Let's talk about the denaturation of these enzymes. And before we can do that, I need to tell you a little bit about proteins. Proteins are like long ribbons. And these long ribbons are created in the ribosomes. And then with the help of the endoplasmic reticulum and sometimes the Golgi apparatus, these long ribbons are folded and twisted and into an elaborate configuration. And you can see here there are curls that can form with this long ribbon. There are loops and they can wind back on top of each other. They can be folded and twisted, and it gets very, very complex. But this protein is functional. And although we've been drawing enzymes as like a round bubble, enzymes look kind of like this. Perhaps this is the active site. Maybe this here is the active site and the substrate would bind right here. I don't really know. I don't even know if this protein is an actual enzyme. But if you were to imagine, this might be, an imaginary active site that a substrate would attach to. But this is actually closer to what they look like than those blobs we've been drawing. Now, what happens when a protein gets denatured, so the temperature is too high or too low, or the pH isn't what it should be, you can actually break the chemical bonds holding these swirls and these folds in place. And when that happens, the ribbon loses all shape and it just becomes like this huge wet spaghetti noodle. And this huge wet spaghetti noodle no longer has an active site. And so it no longer functions. And as far as the body is concerned, it's easier to recycle the pieces of this ribbon than it is to go through the effort and energy required to perhaps refold it. So a denatured protein eventually will be found by a lysosome where it will be broken down into the individual pieces and recycled. So think about why enzymes are necessary for our survival. I've given you lots of examples. See if you can remember those or find some on your own. And I will see you in class.